Okay, so welcome to PHAVES number 13. Crazy, we started this in June of last year and we thought it would be very temporary, but it has turned out to be something that everybody's really loving, so we are going to keep doing it. So we're here and our speaker today is Professor Jon Iredell, and so he is at the Westmead Institute of Medical Research in Sydney, Australia. He's the director of the Center for Infectious Diseases and Microbiology. And so we're really excited to invite Jon because he brings the medical perspective of phage therapy. He's used phages to treat patients down there, and they've been able to show safety with some Staph aureus cocktails in septic patients. So really excited about the work that they've been able to do there. And but also coming in on the research side, he's also a professor at University of Sydney. So we have the science meeting the medical, which is exactly what we need for phage therapy. So really happy to have him here with us. And also we've been working together with their group for putting together an Australian Phage Network and helping that all get set up. So there's tons of reasons why we wanted Jon to come and be one of our guests. So we're gonna hand over the reins to him, and please ask your questions all throughout during the chat, and all those and comments too. I always share the chat with speakers afterwards. So if you wanna say something, so go ahead, Jon, the floor is yours.
Thanks, everyone. I'm just going to share this screen and hopefully won't fumble it too badly. Now hopefully you've got a full slideshow. So listen, thank you very much for the invitation. I'm very grateful for the opportunity to speak to everyone. I hope you can hear me okay. I've got a plane going overhead, but I'm sure Jessica will give me a big signal if I'm fumbling. Look, I was reflecting on this a little bit in preparation for it, and looked at a talk that I gave at ID Week a couple of years ago, and it strikes me then, or strikes me now as it did then, that we're really building on a long, long history. I don't need to tell this audience that, I don't think, but it is humbling to reflect, I think, on the fact that we come at the end of a long line of successful investigators who've contributed a great deal to our modern understanding, but clearly the work is not finished. So what I'm going to do today is talk a little bit about what we're doing here and where we hope to be going. When I looked at this talk, I also reflected on the fact that probably, at least for us, some of the modern momentum was heralded in by papers like this from Chip Schooley and the team, which really got an enormous amount of publicity and made an enormous impact on the scientific and clinical community. And since then, I think there's been exponential growth in interest and quality research output in this area, which is something we should all be grateful for. So my task is to bring you back down under to talk about what we're up to here and where we want to be going. And it's important to note that really the phage therapy story started in 2002 with a chap called Tony Smithyman, who worked with Hubert Mazur, who's actually a distant relative of Felix Durero, he tells me, and Sandra Morales, who is well known in this community. And not long after they'd been going, Tony and colleagues were importing phage from Tbilisi. We treated a patient at Westmead Hospital successfully and subsequently published it. And at that stage, the Australian government's Therapeutic Goods Administration, which is, I guess, our equivalent of the TGA or the European Medicines Agency, didn't know what to do. In fact, told me that I should just sort it out with the drug committee and the ethics committee and not worry about it too much. And so I couldn't really engage them at that stage. Subsequently, Sandra, as you know, moved over to Amplify Biosciences, and we worked with this team who generously provided us with phage and support to do the project. And we're able to study the effect of Staph aureus in people, as Jessica mentioned. And Amplify, of course, subsequently became Armata. At this stage, the regulators said to us, well, We'll put it in through our special access scheme. And I really had pushed this, to be honest. They weren't ready for it. They didn't know how to manage phages, and they treated it as a bit like what the FDA calls an IND. Subsequently, a physician, infectious diseases physician, a paediatrician at the adjacent children's hospital, has started working with us, and she has, Tommy, we can see on the bottom of the screen, she has a bit of a heartwarming story about a young lady we can see on the right who was particularly injured by acquiring a nasty resistant Pseudomonas infection in bone that complicated an orthopedic injury in India, was extremely resistant. And this is an important story because Amini and spoke to us, but we reached out to Phage Directory initially, and Jessica and Jan helped us reach out to the entire community, many of which will be present on this call, to which we are, of course, enormously indebted. And Ran near Paz had a lovely anti-pseudomonal phage, which was in turn made available through the generosity of Adaptive Phage Therapeutics, who had a purified preparation of this, which they made available free of charge to this young lady who was treated by Amine at the hospital and had a terrific outcome, which has received a fair bit of publicity in Australia, in fact. And this is a bit of a poster story for phage therapy here. We did what we normally do in this sort of situation and monitor what we're doing, where the phage is going in places like the bloodstream, so that we've got a bit of an idea about what to do with dosing. We routinely monitor our dosing schedules and adjust them as a result. And we monitor the consequences of the treatment. In this case, we saw an interesting surge of bacterial DNA into the bloodstream soon after therapy began, and that was accompanied by an inflammatory response. And when Ruby examined the transcriptome of this young lady, you could see this, something we had actually seen before in staph patients and in others, this initial innate immune response followed by the adaptive immune response, and this is reflected in antibody development and so forth. This is unpublished at the moment, but Amine's got a preprint up on Research Square. And of course, the other big pathogen in kids and in everyone that is raising a lot of interest in the phage world is, of course, Mycobacterium abscessus. And as you know, Graham Hatfield's team has published a rather spectacular story with a genetically modified mycobacteriophage in Nature Medicine a while ago, which I think many of us know this story very well. And Graham generously made available some phages, including some of the ones that we used then for Amine to treat another young lady with Mycobacterium abscessus infection. And in fact, we've treated a few people with Graham and Rebecca's help since then. So I mention this because it's another example of the kind of interdependencies and collaborations that are so important in this story. For this treatment, as in all the others, we routinely do phage-specific therapeutic monitoring so that we know what's happening with our drug delivery or our phage delivery. We routinely do bacterial monitoring, as I mentioned before, and we routinely test phage antibiotic synergies. Partly it's to make sure that generously provided internationally sourced material survive the trip. That's usually not a problem, but we always check. And partly to make sure that the antibiotics we're co-administering aren't interfering with the therapy, or at least as much as we can infer that from an in vitro testing system. And, there's lots of caveats around that, obviously. And of course, we do the host monitoring when we can. And that's simple things like the clinical markers we all have, but we're still trying to get insights from what the human transcriptome is doing. And that's, I think, still quite informative. All of this, as I mentioned to you before, is subject to the blessing of the regulators. So this is an important distinction in the Australian system, in that our therapeutic goods administration is different from the EMA, it's different from the FDA. And one of the important differences is it actually confines itself to therapeutic goods, which includes biologics and includes drugs, but it doesn't regulate genetically modified organisms, and that is separately handled. So in the instance of managing this story I just described, using Graham's phages, we expected that although the TGA had kind of grudgingly accepted us, because by now we had treated quite a few people. I think we've treated, I don't know, about 18 or so people now with various types of phages for various types of infections. We expected a different reception from the Office of the Gene Technology Regulator. So this is the office in the Department of Health that looks after the risk of people like us releasing something that doesn't exist in nature into the environment for the first time. And fair enough too. Goodness knows we had trouble with the cane toad. So we expected a bit of a grim reception from the OGTA, but was surprised in fact. And they willingly accepted this and gave us a pretty low risk assessment and a license that reflected that. And so Harmony was able to effectively manage the importation from the University of Pittsburgh, from Graham's lab, and we were effectively able to manage the handling of the genetically modified organisms, both at the bedside and in our lab where we did the complementary testing in collaboration with Rebecca and Graham. And similarly, we can do nothing, as I think I've made increasingly clear, without the generosity of industry and academic colleagues around the world. So we've been leaning heavily on advice and support and all the things that we all reach out to in the academic space, to all our colleagues, many of you in fact on this call. And we'd get nothing without the support of our senior colleagues, our academic physicians in the public hospital system, because in many parts of the world, this is really very new, and Australia is no exception. So we have our champions at the Children's Hospital, people like Chris Cowell, and at the Adult Hospital, which is immediately co-located. So Harmony and I work literally side by side in adjacent connected hospitals. People like Jeremy Chapman, and we've really relied on these people to push through red tape at the academic level. So if you're just establishing phage therapy in a clinical environment, you need these champions to help you push through the red tape. Otherwise, it can take you years to get up and running. And of course, in addition to our international colleagues and supporters, as Jessica mentioned, we rely heavily on our organic network, I think, of an open and generous peer group of collaborators. And we hope to coalesce this into this increasingly effective network. But of course, like all of you, we have to start somewhere, and we start with collecting. So here's a photograph of Ruby and Ali digging deep, as it was, to collect samples. And we can do nothing without screening. So this relies on having good quality bacterial collections. And I think this can't be understated. You must have good representative bacterial sets. So we have the luxury where we're located of access to large and well-organized libraries of the bacteria that we're interested in, because we also happen to manage the state and national reference laboratories and surveillance programs, so that it means if you want to ask a question about Australian Klebpneumoniae, in this case, Carola Venturini's looking at the famous subtype of Klebpneumo that gained its notoriety for spreading KPC around the world. And so we're able to make an intelligent assessment of whether a bank of bacteriophages, shown here on the screen, is likely to be effective against any given representative of that particular subtype of Klebsiella pneumoniae, simply by taking representative strains which you've already predetermined, have a good representative position in the genomic population, and taking phenotypes for the phages through a variety of testing methods and inferring a relationship. Increasingly, this kind of work obviously is falling to machine learning and artificial intelligence approaches, but this is a kind of a standard approach, and of course we need to be able to do this to get intelligent assessments about developing good phages and assessing things that look like good phages. Obviously, once you've selected a bunch of phages which you think are good candidates, then we go on to, like everyone, testing, direct testing. And I guess partly because we don't quite trust any method, and partly because we're probably not entirely familiar with every method, we kind of use everything. We use efficiency of plating approaches like everyone because it's quick and it's easy and it's something we're all familiar with. And we use growth curves because, you know, they're also quick, they're easily automated, and all this kind of stuff can be done in high throughput. And we need to be able to do both of these methods, and we do them routinely as we screen banks of phages or libraries of phages against banks of bacterial isolates. And all of these, of course, must be properly characterized before you start to head into this kind of level. Here, Alicia has it's just showing a little bit of data for a screening against Shigella. And the important part of this, and the reason I put it up there, is you can see on the right side of the screen that once we start to take things more seriously, we need to start thinking about our antibiotics. So for a lot of bacteria, we're really treating them with beta-lactam style antibiotics, but for some of the intracellular pathogens like Shigella, we need to be thinking about antibiotics like the quinolones, the macrolides, those kinds of antibiotics. In here, we've got a quinolone, of course, ciprofloxacin, used with different MOIs of the relevant phages in a synergy study. And of course, by the time you're starting to build in different MICs of antibiotics, different MOIs of phages, and a whole bunch of phages against a whole bunch of bacteria, you're creating a lot of work. So you need to be constantly winnowing out things that fail that fail that process along the way. And you really need to head towards automated systems, I think, to do this kind of thing effectively. And some of these things become very labor intensive. For example, Alicia had to move into macrophages to really test what ciprofloxacin and good looking phages do inside a human cell, because of course that's where you're trying to get at the Shigella. When we're treating patients that we've chosen our phages with, I think I pointed out before, we need to do monitoring, and we need to do that because we need to understand whether there's some sort of predictability in this treatment protocols, or whether, you know, we're stuck with therapeutic monitoring as a routine in phage therapy. I don't think I know the answer to that at the moment, but I must admit I'm leaning towards therapeutic monitoring as a routine. And this has important implications for companion diagnostics. So that's something to think about when we're trying to develop phage therapy as a strategy, that the companion diagnostics may be part and parcel. So we try to stick to a routine for all of the patients, irrespective of what kind of phages or bacteria we're using, whether intracellular or extracellular pathogens, that enables us to try and get comparable data. And I'm sure you've heard Jessica and Jan and Shauna and so many other people talk about the need for comparable data and for us to collect these data so that we can all understand what's happening as we go. So I mentioned our colleagues on whom we are so interdependent. Really, we have got now an established network in all the major cities and we are trying to grow this. And if I'm leaving anyone out, it's just my accident and not by design. But essentially what we're trying to do initially is identify the big phage collections, and there's quite a few collections around the country, and I think more and more people participating, and identifying also, in addition to the scientific community, we need to identify those people who've got the capacity and the interest to actually manage the patients with therapeutic phages. And that network is similarly important and needs to be developed in parallel. We've learned a lot from the conversations, particularly from our Belgian colleagues, about the need to have some sort of compatibility when you talk about phages, so that if you have a generous colleague in Israel who provides a fantastic anti-pseudomonal phage that is purified by other generous colleagues in the US, then you need to be able to understand just from a bit of communication about the genome and about the way that phage acts against certain bacteria and the conditions that it acts in and its stability, to know whether you can reliably pick that as a winner before having to import it and test it all yourself and find, oh, gee, that's no good against my strains, or it's no good against, doesn't tolerate the journey or whatever. So there's a lot of work to be done about this kind of area, and that's obviously core business for all of us, I think. The agreed data sets we've talked about, and we also need a way, and I think this again has been something that, you know, Jan and Jessica have championed, and Shauna has talked about a lot, the data sets and the ways in which we might be able to make contributions painless, so that people don't feel like they've lost control of their material when they give it to someone. So we've been learning a lot from other experts who have big biobanks, and trying to understand what a truly collaborative and interconnected network would look like, so that someone can feel comfortable about providing material in a truly participative manner, rather than just handing over their hard-won phages and never seeing them again. I think that's important. We're starting off with trust relationships, and we need to manage them very carefully. So Ruby Lynn has defined here what we think our structures currently look like, or what we think they need to look like. I mentioned before that we have this clinical network, which we sort of define as our therapeutics network, and a source network, which is really, we're talking therapy here. So I'm not downplaying the research side, because clearly, you know, that is so essential at the moment, possibly more than ever before. But I'm here talking about therapy, so I'm talking from the bedside perspective. And so on the green side, we're really talking about the phage access side. And so that's a lot about logistic stuff, as I've already mentioned. And a lot of that logistic stuff is about the legals, it's about the regulators, it's about the governance and the frameworks we need to operationalize all of this. On the therapeutic side, it's about making sure that we know what we're doing. And that means not only correct targeting, so we have the luxury, as I said before, of having a good way to target, because we've got a good grip on our major pathogens here. And I think you need that wherever you're operating, whatever country you're in, you need to pair your work with good quality bacterial sets that you're targeting. And it means understanding companion diagnostics, as I mentioned before. And more importantly, I think, because we haven't touched on this, and I won't speak much about it, but I will highlight it now, and that is the need to do intelligent clinical trials. So at one extreme, you might have the registry concept, and at the other, you might have the adaptive Bayesian trial. And I'll mention this briefly in a second. Clearly, at the business end, we need good quality genomics, we need high throughput testing, we need machine learning and artificial intelligence, and many of you on this call are much more expert than I in many of these areas. We, like you, I think, have the usual public-private tension. the commercial sector needs to make it sustainable. And it's important not to sneer at this. We need to support our industry colleagues who at the moment are acting out of generosity to support what the therapy people are trying to do and support what the research people are trying to do. But we can't kill the goose that's laying the golden egg here. So it's really important that we find ways forward in the commercial sector. This has to be sustainable or this will die. The public sector, on the other hand, I don't know if many of you on the call work in the public health system, but I have all my life, and I'm only interested in making it affordable. And so there is always a tension between the sustainability and the affordability. And that's really the pointy end of that's at the bedside. We have the GMP question in our faces, of course, because some regulators are clearly going to drive you down that path. Other regulators have different registration pathways, and so there's a lot of important questions in this space, and that needs a whole other conversation, I think. And of course, clinicians among us understand the clinician skepticism, and that's really because although there's a lot of anecdotal safety experience, this is really, we've got to remember, this rose in the pre-regulatory era. And so the classical RCT paradigms were not applied and have not really been met. So I'm not sure if I'm covering up my own screen here, I hope not. But we need to be careful that the urgency and demand in the clinical space doesn't disrespect the generosity of scientists and industry colleagues who provide material. I was speaking to some colleagues not long ago who pointed out that they have been generously providing material for people but don't get back the data that was promised as a condition of that provision. So, we need to be careful about burning our bridges here. Now what's happening? Here we go. So where are we here? We in Australia look to be operating in this risk-based frameworks. We've moved some of our major regulators into a more risk-based paradigm, and that's actually going to play well, I think. So the Therapeutic Goods Administration, the TGA, which as I said is our FDA equivalent, they have moved to this now and are more willing to say, look, if you can show us that it's safe and there is a clinical need, we're happy to have a conversation about this. The gene technology regulator has a similar perspective. They will take something that doesn't exist in nature and potentially allow you to release this into the environment as long as you can show that it is for the greater good and it is a safe thing to do. So their assessment is very detailed and something we're experiencing for the first time now. To give you an idea, they will take about a year to assess a new, and this is a first time sort of thing, so a new genetically modified therapeutic. And they will bring to that a group of people with significant expertise. So a postdoctoral team with expertise in genetic modification, expertise in biology to assess this. So they take this very seriously. But once that initial assessment has been made, then it becomes the template for subsequent applications. So as I mentioned to you before, Graham's phages, which had previously been used in humans and previously accepted by the English regulators, went through almost without a hitch through the Australian genetic regulator. And finally, our National Association of Testing Authorities is moving to a risk-based system in order to define things like companion diagnostics. So when we put up an application that includes what we think is a necessary companion diagnostic, the Therapeutic Goods Administration will require it as part of our inherent management system for therapy. and the National Testing Authority will assess it for fitness for purpose. And that's a little bit process-based. In other words, are you guys likely to put your thumb in it and stuff this up? Are you a good lad? Will you do a good job? Can we rely on you? And use that to determine whether it's fit for purpose and will be subsequently licensed for use, including ultimately as a commercial entity. So if I have to do some crystal ball gazing, where do I think this is all heading? I'm hoping that we end up with a risk-based pathway to registration in Australia that emphasises quality control over process control. I think we will probably need a component of GMP, or at least GMP-like systems, because we have to maintain international compatibility. I think we'll end up with a network of phage libraries around Australia, and I hope that we can borrow from our expert colleagues in the Northern Hemisphere in particular to develop some sort of a standardised passport that's compatible with international definitions of a phage that's in a library. I believe we'll have standardised companion diagnostics, and I think the work that's being done at the moment will help inform that. It's going to mean selection processes that's increasingly becoming streamlined, and it means therapeutic monitoring at the bedside as we go, because we have in fact used this for adjusting phage as we go in treatment of patients here at Westmead. And I hope we have a standardised data collection system that is completely compatible with international systems. Now, how that's going to look is a really big and difficult conversation, not one where I think we've got time to tackle today. But the paradigms that come to mind are the registry system, which is really a very passive one and is commonly used by our colleagues in cancer research. So, for example, you will say, look, I want all the Pseudomonas cases treated with phages, lodging a standard set of data and descriptors in a standardized registry, which is accessible for people to study, to mine, to learn from. That would be a registry type concept, such as we understand it. And at the other extreme is the adaptive Bayesian trial, where you will have a multiplicity of approaches to a given problem. Let's say Pseudomonas infection in cystic fibrosis, and you'll have a range of approaches which are intercomparable and will be brought into the trial with a Bayesian analysis of their efficacy as you go, and they'll be dropped in and dropped out. And I think this is likely to be our first approach for cystic fibrosis, for example, here in Australia, the Bayesian adaptive system. But as I said before, we have to make we have to find a way to make this sustainable. So how do we do that? In the public sector, I think we're going to be working on a cost recovery model. So, we're going to be making arguments about the benefits in clinical services, and that means that people who want business cases in the public health sector are going to demand proof that adding the phages in at cost X will reduce the impact on patients and ultimately reduce hospital costs. In the private sector, it's about provision of therapeutic goods as we understand it here. And that means there's intellectual property in the selection of the phages, in their combinations, which phages go with which, and not only just the phages, but of course, which phages with which antibiotics, packaging them together. And of course, the phages as vehicles, whether you have phages that act to deliver an antibiotic to site, or even phages that are truly genetically modified and therefore have unique, they can be patentable as a completely unique entity. That's an obvious way to go. And of course, there's the diagnostics. I think there's going to be intellectual property in here. So this is where I think we can expect to look for sustainability in both the public and private sector. And I think we can satisfy this difficult tension between affordability and sustainability. I'm making the argument, or we're making the argument here, that this is really the third approach. We have vaccines, which have been around for a long time, and they're fantastic. They're highly specific. and they're usually just not toxic at all, but you just can't cover everything with a vaccine. And in fact, truth be told, there's not many diseases that we really have great vaccines for. You know, you think of hepatitis B, you think of smallpox, you think of polio, you know, but a lot of things, they're just really not great. And of course, we've got antibiotics. This is a 60 billion Australian dollar industry, but it's under threat from antimicrobial resistance. It's a bit of a lost leader for a big pharma. So the problem there with low specificity is recognised by everyone. The toxicity problems are recognised by everyone. And so, you know, here comes phages, been around for a long time, I mean, I think most of us would probably argue that there's a phage for every bacteria, very specific. And in the age of precision medicine, which was, you know, this was a disadvantage before to have something that was so specific. But now I think we can argue this is a real advantage. This is the era of precision medicine, and this is the tool of precision medicine. And it just appears to be non-toxic. So I think this is a real winner, and I think this is why the clinicians are starting to embrace it. So we need to just take these arguments forward. Our network is blessed with a good strength of trialists. We have people here who are real expert in clinical trials. You know, people like David Patterson, who really is an international leader in infectious diseases trials. We have fantastic scientists, and we have groups of people who are willing to just work together to make this happen. And it doesn't hurt that we have mutual self-interest. Ruby and I were chatting yesterday with Ian Grange and just pointing out that the need here is met by partners which benefit from their access to biobanks and networks, to making their phages available for the greater good, but retaining rights of prominence over those phages so that, you know, when they're used successfully, when publications arise, because, you know, that's the currency of research, isn't it? That they're duly acknowledged. And the network grows exponentially in resources by these donations, including the data that are derived from users. We can strengthen partner applications so that if a researcher dips into the network as a partner member, draws from that to strengthen a research application, the whole network benefits from the added resources and the research that's done on the material that's in the network. And of course, a strong network attracts government funding, research funding, and it even attracts international partners who want to run trials. So we think of this as a strong ecosystem. And so I want to end with a bit of a plug, because of course, it's a picture of a lovely ecosystem that is the 2024 mascot, if you like, or the emblem for viruses and microbes, which is coming to Australia, we're delighted to say, in 2024. So Portugal next year, Georgia the year after, and then Australia. And of course, in Australia, we'll welcome you to Cairns, which, for those of you who are not familiar with it, is at the top of Australia, which is the bottom of the earth. And it's about there, no matter where you come from, that's still where it is. When you get there, you'll be greeted by friendly locals. We have excellent transport systems. We have ride share like everywhere else. So you can get your Uber, you share with anyone you like. We have, of course, recreational fishing, swimming and diving, which is very popular. But if you want to relax and just take in the wildlife and, you know, maybe do a bit of bird watching, that's also available. So I think I'll close on that, and maybe Jessica will ask if there's any questions. So Jessica, maybe I should stop screen sharing, is that right?
Yep, go for it. That's perfect. Stop screen share.
Okay, I'm back.
Get to our questions, which I've seen a bunch come in. So thank you so much. That was such an awesome talk. And yeah, like, we're so excited about all the stuff going on down there and excited to be part of it. So let's get going with questions from Joseph Michael. Just wondering, which are the major innate immune cells identified during phage treatment at the onset of the treatment and at the end of the treatment?
So the main immune cells, oh, that's a tricky one. Look, what we've been doing, and when I say we, it's really been driven by Ruby Lin, who's the expert in our team on the human transcriptome and the inflammatory response. So we've really been looking at message RNA. And I suspect, in my ignorant clinical manner, that these are coming from the usual immune effectors. So we're seeing stuff that are signatures of things like macrophages. We expect to see a lot of things that are driving down the T cell and the B cell pathway, you know, the usual inflammatory pathways, the pathways that light up down the TLR4 type receptors, which are the kind of things that we would expect to see driven from things like LPS, bacterial products. But we're also seeing, to a lesser extent, stuff from the TLR2 pathways. And that's the sort of thing that might light up when you dump a whole bunch of viruses at the immune system. And I think Ruby's analysis have pointed out that we see our initial inflammatory response as a bit of a mixture, to be honest. You know, we get what you might expect if you chucked a foreign antigen at someone, like a bunch of viruses. We get what you might expect if you give a surge of LPS into the bloodstream. We pointed out before in the staff paper, and even more so, I think, with the Pseudomonas story, that we seem to see a typical TLR4 pathway type response to that follows that LPS surge, which we presume is what we're seeing in company with the surge in genomic, genomic material that matches Pseudomonas DNA in the bloodstream. And then later, at the same time that we see, and I didn't show these data, but there's a little bit of data that matches what everyone else is finding, which is that, you know, you're getting your IgG, your IgM coming up about that, you know, day 7, 10, 14, just like you expect with everything else, really. And that you're seeing the transcriptional markers that match that kind of response. And I think I flashed up a little in the corner of my screen, one of Ruby's little diagrams that showed that crossover between that initial, you know, innate response when you're dealing with the infection and that surge into the bloodstream that we think we saw, and then that sort of switch over to the adaptive response at the end of the week. And it's kind of complex because, you know, you're still dialing in this foreign antigen in the form of virus, but it's antigen that is not always that foreign. When we looked at our staph patients, we saw people clearly had experienced these antigens because of you looked at their IgG and IgM, and it's unsurprising that if someone's being treated for Staph aureus with, you know, the kind of phages that we know are all pretty related, they're going to have met those phages before or something like them. So that was a waffly response. So I hope I got close to your answer, but yeah, I'm not an immunologist, as you can probably tell.
Thank you. Yeah, well, that was super interesting. Me at least. From IL Hardy, when studying phage antibiotic combinations in vitro, did you observe cases of antagonism between phages and antibiotics? Did the antibiotics ever decrease the efficacy of phage treatment?
Oh yeah, absolutely. I didn't show you any of this, but in vitro, and I think this is well established, there's plenty of published material out there. You know, clearly if you use an antibiotic that's going to act in such a manner as to interfere with things like receptors or basic mechanisms, of course you're going to expect antagonism. I think more commonly we probably see nothing much or a bit of synergy. So if we see, you know, strong antagonism, that'll put us off the antibiotic. And I think in our experience, though, mostly we are seeing just a bit of synergy or nothing. And sometimes we see quite strong synergy. The trouble, of course, and many of you on the call would be aware of this, is that we're all a little bit skeptical, let's just say healthy skepticism, reserved judgment, about the correlation between the in vitro data and the in vivo effects. So if you see an MIC, and we think we trust that as a guide to treatment, we know that in fact, in the real world, that MIC is not the whole story. There's lots of other important biology going on that determines outcome. And that's just talking about a really well understood antibiotic like a penicillin and a really well understood bacteria like an E. coli. And then if you start chucking a second drug in and you try and understand the interaction between those drugs and then infer the in vitro findings to in vivo, that's getting much more complicated. No one's quite sure what that means. So much so that in fact, very few laboratories offer synergy testing as a routine. We do in our laboratory, our service lab, but most other labs in Australia will not offer synergy testing. And I'm just talking about common antibiotics like a beta-lactam and the aminoglycoside. And if you want to put phage synergy in, an antibiotic phage synergy in, you really have to sort of suspend disbelief. But we're doing it because we think we need to learn from it.
Got it. Awesome. Thank you so much. This is from Sylvia Worsley. Thank you so much for this great lecture. Sorry if I missed this. May I ask in which intervals you monitor bacterial genome copies during phage treatment? Same intervals as CRP and leukocyte count values?
So what was the question about? I think I just had trouble hearing.
Which intervals do you monitor the bacterial genome copies during phage treatment?
Yes, sorry. Yes, the same intervals. It's just a routine. I won't share screen and go back to the slide, but we're trying to stick to the same routine we set out with, which we put in the sort of descriptor of the staff story. So we think there's a lot happening in the first few days. So we kind of have a bit of a focus on there. We try and do a bit of an early kinetics because we think people's clearance may differ a little, but in fact, it's turning out to be a bit the same. So we're getting some sort of a reticuloendothelial style clearance of viruses from the blood, but we check that in everyone. And so we'll repeat that late because we think the antibodies start to interfere after a while. But the rest of the time, we just do everything. And it's kind of day 1, day 3, day 5, day 7. I think it's 10 or 14, and then later on. And we usually see extinction of the bacterial DNA in most sort of circumstances within that first week, whether they're bacteremic to start with or not. Yeah.
Wow. From Ian Grange, with respect to genomic screening during and following phage therapy, have you done this to the extent of looking for mutations present in bacterial infections that persist after therapy that might be phage resistant, or phage mutations that take over a population?
Yeah, absolutely. Yeah, and I think most of us would regard that as mandatory, really. I think even our regulator, when they're saying, yes, you can use phage therapy, they're saying you better be looking for resistance. So we routinely go after anything we can culture of the organism that we started off attacking. In fact, mostly we have experienced extinction from major sites like the bloodstream or wherever we're going after, but also from, carriage sites. And in fact, we have not seen resistance yet in vivo, presumably because by the time you've dealt with, a bunch of antibiotics, a host immune system, iron starvation, oxidative stress, nutritional depletion, a bunch of bacteriophages, you just can't make it through to the other end. So I think the fitness costs of this assault on bacteria is really preventing effective adaptation. It's easy to do it in a test tube when all they've got to do is kind of manage the phage and maybe an antibiotic, but it's much harder in vivo and we're not seeing it. We haven't seen it so far. It'll arise, of course, but yeah, we certainly go after it.
Got it. Here's another one on phage antibiotic synergy from Vuong Li. Regarding phage antibiotic synergy, what is the underlying mechanism behind such interaction? Is this a common phenomenon or specific to the combination of phage and antibiotic class?
That's a really good question, and I really don't know the answer, and we're really working hard on it because it's really interesting. So if you can give us any clues, I'd love to know. But yeah, I mean, I think we would all just infer a few obvious mechanisms, I guess, wouldn't we? If you're, I guess at the crudest term, if you're forced to make a trade-off between one fitness cost and another, and that fitness cost disadvantages you in your capacity to make a second trade-off, then I guess that's what our expert colleagues in in this kind of area would say would be the typical mechanism. So what those trade-offs are is the key question. We're really interested in bacterial stress responses. I wouldn't claim any huge expertise. There are many probably on the call who are more expert than we are, but nevertheless, I think that's where the guts of this story is. The trouble is that the study of trade-offs in the Eppendorf are not going to be the same as the study of trade-offs in the humans. So getting an animal model and studying trade-offs inside the animal model is very difficult. You don't always have a good animal model. Trying it inside a cell is not a bad surrogate. I think that is a really exciting area to be working in, and a really difficult one. But yeah, I'm really keen on it.
Oh, there's so many great questions. Yeah.
That was avoiding the question, that one, so I'm sorry about that.
I'm not really judging you on those. I just go to the next. So you can skate no matter what you want. Okay, we got a couple more. I'll do 3 more questions, and then we're going to do a 5-minute breakout so everybody has a chance if they want to meet somebody new. So here's first of the 3 from Paul Jaschke. The genetic modifications of the Hatfill phage are quite modest. Do you have a sense for how OGTR would deal with something more substantial, like a gene from a different phage?
Yeah, that's a very good question, Paul. So as I said, they have moved to a risk-based assessment and they take it very seriously. So they would look at the modification and say, what does this do to its biology? And they have people who are competent to make those assessments. And so a dramatically modified phage would really, I mean, most of us, when you think about the engineering that we're likely to be wanting and likely to be doing, they're going to be things that are going to promote specificity at target. They're not going to be things that are likely to drive a problem in the environment. But if someone were to try and make a cure-all phage that had very broad specificity, I think our regulator would object to that. They would say, look, we really can't let you, I understand what you're trying to do at the bedside, but we can't let you release an organism that doesn't exist in nature that's going to go out into our oceans and kill everything it sees. So that kind of stuff is going to be a realistic block. So I think, the engineering of highly specific phage is not going to be a problem. The engineering of something that's likely to make a mess outside is going to be a problem.
Got it. Awesome. Okay, 2 more from Edison Kano. Is that LPS response you're describing from biomarkers or clinically just like sepsis? Have you seen hypotension or shock with IV phage administration?
Yeah, another good question. So I remember reading Durrell's published lecture from 1931. He talked about injecting people's staph lesions with filtrates and how they all got this big fever a few hours later and all felt terribly unwell, and then they all got a bit better. And I remember thinking at the time, you're just squirting in a bunch of LPS into these poor devils, and so no wonder they got an inflammatory response. But obviously the material we've been using has been material that meets the FDA specifications for LPS and the European specifications for a tolerable amount of LPS. And so you're not really injecting a pyrogen at the level that you're expected to drive a fever. Obviously, you can push the boundaries a little bit and get close to it. You might get a bit of a tickle of fever, but we don't think we're seeing so much a response to that as we are seeing a response to the LPS surge that accompanied the lysis of Pseudomonas in the case that we mentioned there. So in our critically ill patients, this is the staph group, we had a bunch of people on life support, on maximal inotropic support, so their blood pressure was all artificial. If you took all the drugs, though, they wouldn't have any. So it's very hard to tell in that community. In the more well people where you might see such a response, we got a surge of DNA into the bloodstream that immediately preceded a fever, pain at site, no hypotension, a jump in the classic markers like the C-reactive protein, the procalcitonin, a white cell count jump by the next day, and then it all kind of settled. And that was in the Pseudomonas case that Arminet treated that I referred to, which is just on Research Square at the moment as a preprint. And then because we saw presumably less amplifications from the population of bacteria as it subsided, and possibly because antibodies were starting to emerge, we actually increased the dose in this young lady, and we got a smaller and similar response. Not as big a surge of DNA and a less of an inflammatory response. But we think this was probably a response to the bacterial pyrogen, not to the administration of the phage prep itself. The phage prep was provided by APT, and we think it is a very clean phage.
Awesome. Okay, last question, then we're going to break out into our breakout rooms if people have a few extra minutes. So from Greg Canfield, do you find some sites of infection and colonization more difficult to treat than others? For example, is it more difficult to achieve bacterial eradication from the GI tract, a non-sterile site, or the bloodstream, a sterile site?
Yeah, no, that's another really good question. Of course, biofilm infection, you know, for scientists, we all know this is kind of normal, kind of the normal state for bacteria, as opposed to this ridiculous vegetative growth state that we see only in laboratories, really. And we think we see occasionally in infection, but I think all the infectious diseases physicians like myself and Armini and many on the call probably understand that it's the biofilms that are the curse. And so, yeah, we can clean the bloodstream easily. Organisms are very vulnerable in that position. You can kill them with almost anything. You know, as I often say to my trainees, a glass of water and an aspirin will fix that. But in a biofilm infection wrapped around a heart valve, a prosthetic hip, you know, these are a curse, and the antibiotics routinely fail, especially antibiotics that work on high turnover. So the beta-lactams are a dead loss really in that environment. And the aminoglycosides are not much chop either. So this is where I think phages have a bit of an advantage, but of course the biofilm is also a tricky state for them, and we know mechanisms that can block them. You know, the famous story about Curly of E. coli. We know that many of them will be enriched into sister states and various types of growth forms that are really not going to be that vulnerable to external attack, including phages. But I think there is room for optimism that this is the area that we can start to target phages. But I think we need to be very careful about understanding the biology here and not infer from vegetative state phage activity to biofilm state phage activity. So we don't have at the moment a really good mechanism for high throughput biofilm study of phage susceptibility. You know, we have to some extent automated antibiotic susceptibility in biofilms, but it's not widely used. Nobody's quite sure what it means. And it's been kind of abandoned in the clinical space as an indication for therapy. So I think that's a work in progress. So the short answer to you is yes, it's the way to go. And as to whether I know how to do it or have a great system for doing it, no, but that's an area we need to be working in. Yeah.
Awesome. Thank you so much. So this is closing out the formal part of the session, I would say. I'm gonna stop the recording right after this. So the breakout rooms won't be recorded. You can just kind of introduce yourself, say why you're interested in phage, and you'll hopefully meet a couple new faces. So, but thanks everyone for coming. If you have to leave, it's now the top of the hour, so you're welcome to. And you'll find we'll put the recording on YouTube, and we'll talk about it in our newsletter. So if you're heading out, thank you so much for coming. So here we go. Stop.
Yeah, thanks everyone.